Dear students, in this session I would like to discuss on chemotherapeutic agents as inhibitor of bacterial growth. As all of you know, various chemical substances are used in the treatment of several diseases. The treatment of a disease with a chemical compound is known as chemotherapy, and the chemical agents used to treat the disease are called chemotherapeutic agents. Such agents destroy pathogenic microbes or inhibit their growth within host cell. Chemotherapeutic agents can be divided into three main types on the basis of their origin. They may be natural, synthetic, or semi-synthetic. Now let us have a look on the development of chemotherapy. Paul Ehrlich was the first to use chemical compound in the treatment of disease, and that's why he is also known as father of chemotherapy. After Paul Ehrlich, Domag and others have also invented a number of chemotherapeutic agents that can be used in the treatment of bacterial diseases. The first antibiotic to be discovered was penicillin, and that was discovered by Alexander Fleming in 1928. He found that contaminated plate was having the growth of fungus Penicillium notatum, which produced antibiotic penicillin. With this historical background, now let us have a look on the properties of chemotherapeutic agents. To be used as a chemotherapeutic agents, the agents must have the following properties: selective toxicity, it should not harm the host and should destroy the pathogen. They should have a good penetration power, should not affect the host's natural defense mechanism, should have a broad spectrum of activity, should not have any undesirable side effects on the host cell, it should not eliminate the normal microbial flora of the host cell, and they should prevent the development of drug resistance in the pathogens. Now I will discuss about the mechanism of their action. The way they act on the pathogenic microbial cell is called its mode of action. They generally act on an important microbial structure or function that usually differs from host cell. Thus it has minimum adverse effect on the host cell, and that is their selective toxicity. There are 5 different types of modes of action of chemotherapeutic agents: inhibition of cell wall synthesis, disruption of cell membrane, inhibition of protein synthesis, inhibition of nucleic acid synthesis, and action as antimetabolite. First I will discuss about the inhibition of cell wall synthesis. Antibiotics like penicillin and cephalosporin, they are the beta-lactam antibiotic. They inhibit the cell wall synthesis by interfering with the transpeptidation reaction in the peptidoglycan synthesis. Penicillin V and G are the examples of natural penicillin. Certain polypeptide antibiotic such as polymyxin act as a detergents and distort the bacterial cell membranes. Antibiotics like streptomycin, tetracycline, erythromycin, chloramphenicol, they inhibit the protein synthesis in bacteria without damaging the animal cells. Chemotherapeutic agents like rifamycin, metronidazole, and quinolones, they inhibit the nucleic acid synthesis by binding with DNA or RNA polymerases. The last but not least is acting as antimetabolite. Sulfonamides or sulfur drugs, they act as a normal substrate analog and inhibit the growth of bacteria by interfering with their metabolic reactions. Sulfonamides or sulfur drugs, they compete with para-aminobenzoic acid and inhibits folic acid synthesis in bacteria. Thus from above discussion, it can be concluded that the chemotherapeutic agents, they selectively inhibit the growth of bacteria and can be used in the treatment of bacterial diseases. I am very much thankful to BISEC and Arts and Science College Petlat for providing me an opportunity to participate in the state level workshop on e-content development. Thank you all.